So I'm Nicholas. I'm from the Center of Integrated Ecology at Deakin University. My work focuses on the Tasmanian Devil and their transmissible cancer. And I'm in the second year of my PhD. So the Tasmanian Devil is an iconic Australian species, as well as an ecologically important species as an apex predator. It helps maintain trophic levels. Also in the Tasmanian ecosystems, its presence is supposed to help deter feral cats from hunting native species. But I think from the previous presentation, it seems like the devils are lacking in the Tarkine region. They're slacking and need to pick up their work on deterring the cats. But yeah, that's why they're important in those regions. However, over the past few decades, the numbers have been declining. This obviously threatens them, but also the ecosystems they help maintain. This is due to the devil facial tumor disease, or DFTD. The graph on the left shows the population decline over time, and their current numbers are estimated to be roughly 15 to 25,000. Devil facial tumor disease is a transmissible cancer. This is basically defined by the fact that the cancer cells are able to get into and infect another host rather than just maintaining the infection in the person it derived in. And DFTD encompasses two transmissible cancers that have independently emerged, and these are DFT1 and DFT2. The reason why transmissible cancer is such a problem for devils is their biting behavior, which is quite prevalent for the devils. They bite around carcasses and during mating. So this helps sort of transmit the cancer quite frequently, which is why it's been prevalent for so many years. But also, not only does obviously cancer kills them, these large tumors, which typically occur around the face, will sort of inhibit them from eating if they get really large, but they can also become, and their tumors themselves can susceptible to secondary bacterial infections. So as I mentioned before, there's two transmissible cancers, DFT1 and DFT2. DFT1 was the first cancer to emerge, and it emerged in 1996 from a female devil. And DFT2 came from a male devil, which is the sex difference is how we know that they are independently emerged, and DFT2 isn't just another subtype of DFT1. Thus far, DFT2 has been localized to the Channel region, just down here. It is slowly spreading. How far and how fast it's going to spread is unclear at the moment. We still need to do more work on that, but it could potentially spread as far as DFT1 has spread. In terms of the host response of the devils, it's not looking good. There's a reason why it's been so damaging for their populations. Very few devils show regression. Even some don't show response at all, or an immune response at all, which is absolutely crazy for having a foreign cancer cell in your body and just nothing happening, which is ridiculous. And on the sort of cure front of the cancer, vaccines have shown minimal protection in terms of getting the cancer, as well as treating it and showing regression. There is some response with the vaccine, but it is minimal. So our conservation efforts are mostly focused on two areas, obviously overcoming the cancer. This will be in the way of maybe trying to see if the devils can develop natural immunity, or see if we can develop vaccines that enable the host to recognize that the DFTD is very much different from the host cells, but DFTD is very good at escaping that immune response, which we're still not 100% sure, but we have a few ideas on how it does that. The other part of conservation is insurance populations. This encompasses zoos and mainland populations. Like recently, Chris Hemsworth was seen showing working in one of these mainland conservation areas. These are sort of pretty much wild areas on the mainland, as well as the most famous, the Maria Island population off the coast of Tasmania. My work will focus on these two main goals, the overcoming the cancer and insurance populations. With the two objectives, of course. I will be comparing, objective one is to compare the host response of DFT1 versus DFT2. And my second objective is to look at the difference in the immune gene expression and the immune activity of captive versus wild animals. We would expect in captivity from previous research on different species that their immune system is a bit lower in captivity, with that having potential implications for their release into DFTD infected areas. And the overall aim is to improve these conservation efforts relating to those two previous main goals, defeating the cancer and the insurance populations. Both objectives follow similar methods. I'll be looking at immune gene expression for this comparison. They both start off with blood samples, so we're looking at blood samples, which is followed by RNA extraction and then measuring the gene expression. I'll be measuring this using custom panel analysis. This basically is just, I will pick a large range of genes to look at. And then they'll be split at the data analysis section for those two relative objectives. So why look at DFTD versus why look at the two sublineages? The differences may influence the effectiveness of the vaccines, as well as the spread of the disease. So it's important to know this for the epidemiology and monitoring the spread, because that could be different for each lineage. I'll be getting my samples of infected hosts from this blue region, the blue circle here, which is the channel region. I will also be getting my wild samples for the captive versus wild comparison from this region as well. And the aim there is to inform vaccine development and expand knowledge on devil eco-immunology and our knowledge of the cancers. So what would I expect from this? So there's a few differences with DFT1 versus 2. One of the most notable is DFT2 still expresses MHC. This is the major histocompatibility complex. This is the main protein in tissue transplants that will cause a rejection. So given that MHC is still expressed in DFT2, we'd expect it to be more immunology, like it would give a stronger immune response than DFT1, as well as the fact that it originated in a male devil. It'll be expressing male-specific proteins, which should have more trouble infecting females, so that sort of those areas, they might be a difference. The cancers also originate from slightly different cells. DFT1's myelating Schwann cell, DFT2 is repair Schwann cell. The repair Schwann cell has greater capacity to modulate the immune system, so it might benefit from that. The cancer might benefit from that. It's also much younger, so perhaps hasn't acquired as many mutations over time, which may make it more favorable to infect devils and make it more viable as a parasite. For the captive versus wild, captivity is a very different environment from the wild. The animal's much more pampered. For the devils, their food is treated for parasites and they're treated for parasites as well, which is very different for a species that is eating rotting carcasses. Quite often they're not exposed to many diseases they probably would be. And for captive release programs, there's a paper that's showing that disease is sort of one of the causes of death for captive-born animals compared to wild. So disease for releasing animals is something you want to be aware of, especially the devils, releasing them from captivity where they haven't had the cancer into the cancer environments, seeing if the adaptive immune system is lower in captivity, and we need to address that. This is a possible question that I want to try and answer with this objective. My sampling sites are Monado Zoo and Healesville Sanctuary, and these are intense captive sites, so they're the most relevant for this. And this will help inform animal husbandry and release programs, as well as our knowledge of DFTD. Many people to thank for this project, particularly Zoos Victoria and UTAS, but also, of course, Australian Wildlife Society and the Holsworth. Thank you for listening.